Okay, lecture 7, we'll talk about the transcription process. Okay, so last time we talked about the DNA replication process. So first of all, let's talk about the differences between the DNA replication and the transcription process. So, of course, the enzyme that's involved in the processes are different. DNA transcription is mediated by our DNA polymerase, of course, but the transcription process is mediated by the RNA polymerase. So there are two different enzymes. Okay. If you compare their mechanisms, of course, DNA polymerase cannot start the DNA replication without the help of a RNA polymerase that can make RNA primer. But RNA polymerase can start its work by itself. There's no need for synthesizing RNA primer. So RNA polymerase can be more independent, of course. And then, DNA polymerase replicates both template strains semi-conservatively, right? But the transcription is only happening one of the two DNA strand. So one of the two DNA strand, which is a private coding strand, is transcribed. So that's different. Okay. And then the accuracy-wise, transcription is less accurate than DNA replication. Evolutionary, of course, that makes sense because the DNA replication needs to be very accurate, because if it's not accurate, of course, you will get a mutation in your genome. But transcription, in terms of what happens if you make a mistake, the mRNA or whatever the RNA product that's transcribed, it's not going to be passed down to the next generation. So it's more forgiving for the mistakes the RNA polymerase can make. And then transcription is done on a selective part of the genome compared to DNA replication, which is done throughout the genome. So the entire genome is replicated, but transcription is only replicated on a functional part of the genome, which means a gene. A single gene is replicated. Okay, so that's a huge difference. So understand, replication happens on one of the two strands of the DNA template, which is a coding strand, and then it happens even on that specific segment of that coding strand. That will be a functional coding unit, which is a gene. So the transcription is a gene-specific process. Okay. So again, transcription is considered as the beginning of a gene expression process. So when you say a gene is expressed, that means that the genetic information that is embedded within a gene is used to develop a product. That product could be a protein, that's how we learned, or that product could be an RNA if that RNA is never translated into a protein. Okay, so whichever that is, and we consider that as a gene expression product to develop a final product out of genetic information embedded on your DNA gene. Okay, and then the entire chromosome is not transcribed. Like I said, that's a big difference between the transcription and the replication process. And the regulation of gene expression, what it means is then how do you know which gene is transcribed or which gene is not transcribed? Okay, that is the regulation of gene expression we're talking about. Okay, there may be a signal that comes into the cell that decides what genes to be turned on, because they'll work on the transcription factors, which will be activated, and then those transcription factors will be binding on the promoter sequence and then start interacting with RNA polymerase that can turn on the transcription. So that's considered the gene regulation mechanism, which determines which gene should be started, transcribed, or the transcription should be stopped at the end. Okay, and usually the term transcription is used to describe the making of mRNA. Okay, that's the first step in making a protein if it is translated after that. However, we could be talking about making tRNA, ribosomal RNA, and other RNA molecules like ribosomes and things like that, which are never translated. Okay, so, but still transcription happens. Okay, so that may be the gene expression right there until the making of RNA. Okay, so if you consider how many RNA molecules we have in the cell, the living cell, Okay. Then we have a whole bunch of RNA molecules. So these are probably different names of RNA molecules that are serving different functions. Look at this, ribosomal RNA. There are a whole bunch of them. Okay, 18S, 28S, 5.8S, 5S RNA molecules, and there are a bunch of tRNA molecules that are involved in the translation process. And then some messenger RNA molecules are there. And then there are other RNA molecules called heterogeneous nuclear RNA molecules. Some of them may be a precursor of mRNA, and there are other group of RNA molecules called small nuclear RNA molecules, snRNA molecules. There are so many different types of those. And then small cytoplasmic RNA molecules. Okay, what is shown is there are a whole bunch of RNA molecules that are not mRNA. Okay, mRNA molecules are used as a blueprint to build proteins, but the majority of RNA are something else than mRNA molecules. So it's about 90% of RNA molecules are something else than mRNA molecules, which means they will never be translated. Okay, understand that? So don't assume that RNA molecules are all translated into protein. That is not the case. Maybe 9 out of 10, they will never be translated. Okay. And then the enzymes that are doing the transcription. So so-called RNA polymerase, if you look at prokaryotic cells, there is a single RNA polymerase doing the transcription. Okay, so bacterial cells have one single enzyme that is made up with all these different units. Okay, beta, beta prime, alpha, 1 alpha 2, epsilon, and things like that. And then our archaebacterial cells have another single RNA polymerase that are made up with different subunits. But if you look at eukaryotic cells, they have multiple RNA polymerases, 1, 2, 3, and 4. Okay, and different polymerases in the eukaryotic cells, they transcribe different genes to make different RNA molecules. For example, the mRNA molecules you're familiar with, they are transcribed by the enzyme called the polymerase number 2. Okay, but if you're talking about maybe ribosomal RNA or transfer RNA or other RNAs, and they are transcribed by polymerase 1 or polymerase 3 or polymerase 4 and 5. Okay, so understand eukaryotic cells have different RNA polymerase enzymes that are transcribing different RNA molecules. And these are the structures of RNA polymerase enzymes. So, bacterial RNA polymerase, which is a single enzyme in the cells, have these subunits. Okay, but eukaryotic cell, there are different multiple ones. But if you look at polymerase number 2, may have these different subunits. Okay, understand? You don't have to memorize all these. Understand there are complex subunits in each RNA polymerase enzymes. So transcription process itself is very complex in how it's initiated and regulated and elongated and then terminated. You have to understand all those. Okay, so how it is initiated, it's very accurately controlled. Like I said, there's so-called promoter sequence, DNA sequence ahead of the gene coding sequence. Okay, that is probably bound by transcription factors, and then RNA polymerase, and then RNA polymerase is activated to start the transcription. Okay, so binding of those proteins and RNA polymerases are important, of course, and then how RNA polymerase starts to open up the DNA template strand and start making copies, RNA copies up of the DNA. And then once that's done, about at the end of the gene, okay, there may be a specific sequence that will allow RNA polymerase to recognize, okay, it's the end of the gene sequence. I should stop the transcription, and then this engages from the DNA template strand, so it follows up. So that will be the entire process. And then the beginning of the transcription process, if you look at the bacterial cells, normally they have a unique promoter sequence ahead of the gene sequence. If you look at the DNA segment, +1 means that's the start of the gene coding sequence. Okay, and then ahead of that, gene coding sequence, you have some meaningful sequence ahead of it. We designate those sequence region as maybe negative numbers. Negative 1 means right here ahead of the gene coding sequence, 1 nucleotide away upstream. Negative 10 means maybe 10 nucleotide upstream of the gene coding sequence. Minus 35 is minus 35 nucleotide away from the gene coding sequence. That's what they mean. So around those areas, there are meaningful sequences that are recognized by transcription factors. In the bacterial cells, there are transcription factors called sigma factors. Okay, so especially like sigma 70. 70 means the size of the protein, 70 kilodalton protein. Sigma factor can recognize these promoter sequences. Okay, and especially minus 35, minus 10 sequence area are very important. Okay, and then they bind there, and they open up that area. Okay, and to start the transcription. So it's very important. So those sequences minus 10, minus 35 are very conserved, meaning they are found from most of the genes that are initiated by the sigma factors, the same sequences. Okay. So the sigma factor 70 is very important, and this is the sigma factor structure. It has unique kind of regions, maybe 1, 2, 3, 4 regions that have unique amino acid sequences, and they have unique domains that are interacting with the promoter sequence of the DNA and that they recognize and bind. Okay, so it says the 4 regions, 1, 2, 3, 4, and among those regions, regions 2 and 4 binds with minus 10 and minus 35 elements of the promoter sequence. Okay, this region and this region. Okay, are binding with minus 10, minus 35, that's what it says. The region 4 has helix-turn helix motif, as we learned about DNA recognition motifs, right? So helix-turn helix was one of them. So that region 4 has that. Okay, that's how it can bind or recognize and bind minus 35 sequence area. And region 2 has also alpha helix interacting with minus 10 area. Okay, so it's going to melt up that area. And then region 1 has some amino acid aromatic structure like tyrosine, tryptophan, phenylalanine kind of things. Okay, and then those amino acids can stabilize the melted DNA by interacting with the bases. Okay, so different regions of the sigma factor do unique roles in promoter sequence binding. That's what it's saying. And then once sigma factor binds, of course, the DNA RNA polymerase is start working with the promoter sequence. Okay, so if this is a complex RNA polymerase sequence, it binds with the promoter sequence as well. Okay, so the melting of DNA happens, especially between the minus 10 and plus 2 region. So plus 2 region is probably somewhere down there, and minus 10 and minus 11 is somewhere down there, minus 11, and plus 2 is somewhere down there, that area. Okay, so melting happened. Melting means the hydrogen bonds between the two DNA strands are broken up. That's what melting is. So DNA is unzipping. You look at the sequence there. It's a lot of A's and T's. So it's a very AT-rich sequence. That's why it's easily broken up. So the minus 10 area of the promoter sequence. Okay, so especially around that area, minus 10 area, so which is like 11 and 7. So 11 is right there, and 7 right there. Those are important because they kind of flip out and they bind so with the DNA. So there is that minus is that 11 and maybe T7 right there. So they go into the pockets of the RNA polymerase. They flip out, and then pockets within the, I guess, sigma factor. That's what is showing up there, sigma factor protein. So that's a sigma factor showing up. Okay, so they bind, and then that's starting of the melting. So again, here the sigma factor showing up, interacting with those two nucleotides because they have the right pockets for those. Okay.